Hi all, Chetana here, welcoming you to Brilliant Botany. Today I am before you with a very new topic called bioinformatics. Quite difficult, is it? Don't worry, I am here to make it very easy and simple. Okay, in this video we'll be studying what is this bioinformatics. Okay, history of bioinformatics, database sequences, database management tools that we are using in the bioinformatics. Okay, protein sequences and nucleic acid sequences and so many. Especially students from Tumkur University do not study this bioinformatics as a special subject. Instead, they have this bioinformatics as a subtopic under a special paper called as plant biotechnology. So for that reason, it is very much necessary to focus on role of this bioinformatics in the field of plant biotechnology. Okay, we'll study that also. Without wasting time, let's move on to the topic directly. Okay, what is this bioinformatics then? See, bioinformatics is a science which is concerned with the development and as well as the application of computer hardware and software. Okay, this Human Genome Project was completed with the usage of this bioinformatics. Okay, and it was completed by Professor Francis Collins, who is currently the director in the National Institute of Health, Bethesda, USA. Okay, so this is a small introduction, a very brief introduction regarding the bioinformatics. That means I told you about the definition, that's all. But now we'll see the history of bioinformatics. Okay, if you go back to the history of bioinformatics, in the year of 1960, high-level protein sequences was used for managing the protein sequences. Okay, and Margaret O'Day, she was called as pioneer in the field of bioinformatics, and she was father and as well as mother of the bioinformatics. Okay, she collected entire protein sequences in a book called as Atlas of Protein Sequences and Structure and got it published in the year of 1965. So this book, the Atlas of Protein Sequence and Structure, led to the Protein Information Resource Database of protein sequences, which could be accessed by the telephone, and directly it is applicable for the information through the remote computers. The term biotechnology was first used in the year 1970 in order to study the information sources that are taking place in the biological system by Pauline Hogwig and Ben Hesper. Now we'll study in depth about this bioinformatics. Okay, yes, bioinformatics is the analysis of the biological information with the use of computer and as well as statistical techniques. You can say that bioinformatics, it is a science which uses computer and as well as algorithms to accelerate and to enhance the research. Okay, so bioinformatics also deals with the computer science, mathematics, statistics, genetics, biotechnology, and as well as structural biology. Also, this bioinformatics mainly deals with the computational management and analysis of the biological information. Okay, so this biological information will be in the form of genes, genomes, proteins, ecological systems, artificial intelligence, medical informations, and robots. Okay, so yes, this bioinformatics has its major application in the field of protein biology and as well as structural biology, and it mainly works on the properties and as well as the evolution of genes and as well as proteins. Okay, and the process of study of genes, that means the process of study of properties and as well as the evolution of genes with the help of bioinformatics is called as genomics. The same thing if the study of or the process of study of the evolution and as well as the properties of proteins with the usage of bioinformatics is called as proteomics. Now let's move on to the tools used in bioinformatics. Before knowing those tools, let us know how these tools have been actually used. Okay, so computational tools have been routinely used for the characterization of genes, determination of gene structure, gene physiological properties, and so on. So for all the different purpose, we have different kinds of tools in field of bioinformatics. Okay, so say for example, if you want to know the physicochemical property of a gene, so different sequences are analyzed based on the composition of the different amino acids, and based on that you can derive the large informations like hydrophobicity, hydrophilicity, bulk value, molecular mass value, and as well as pi value. Now we'll see the tools that are used for the analysis at the different levels. In the field of bioinformatics, we have two categories majorly. So one is standard bioinformatics tools and another one is customized bioinformatic tools. So these in order to meet the requirements at the different project levels. Okay, so there are different data mining sequences or data mining softwares in order to retrieve the information from the genomics. Also we have visualization tools in order to retrieve the information from the proteomics. Now let's see the databases that are used as tools in bioinformatics. Firstly, SwissPot. So this SwissPot, which is a database which is used as a tool to get information about the protein sequences. Secondly, EMBL. This gives the information about the gene. Then Entrus, it has collection of genes. GDB, it is called as gene database. And Gene Bank, so it has the information about the gene sequences. And finally, GSDB and PDB. GSDB is the gene sequence database, whereas PDB is the protein database. GSDB gives you the information about the gene sequences and as well as gene structure, whereas PDB gives the information about the protein structure and protein database sequences. Now let's see the classical tools used in bioinformatics. Firstly, for the purpose of database interfaces, we have tools like GeneBank, EMBL, DDBJ, Midline, SwissPot and PDB, which means protein database. And for the purpose of sequence alignment, we have BLAST tools, FASTA tools, Clustal, Multi-Align, D-Align. And thirdly, structure prediction. See, suppose if you have protein sequence and you want to predict the protein structure, we have Swiss modeler for that purpose. Okay, and for gene finding, we have GeneScan, GenomeScan, GeneMark and Grail. See, the other group of tools that represent the protein domain analysis and identification is the Grail. For example, if you have protein sequence and if you want to know the domain, location and its location, we have tools like PFAM, Blocks, Prodom. And for the purpose of pattern identification or pattern categorization, we have Gibbs Sampler, Align ACE and ME and ME. See, for the purpose of protein folding prediction, we have tools like Predict Protein and Swiss Modeler. Here is a brief explanation for few of this. Every day this bioinformatics is running with the sequence search programs like BLAST and sequence analysis programs like Emboss and Stadin packages. Okay, and also structure prediction programs like Threadar and as well as matching, and for the purpose of modeling programs we have RASMOL and WhatIf. Now let's see what is this homology and similarity tools. Okay, this homology and as well as similarity tools are related by divergence from the common ancestors. Hence, this can be used to identify the similarity between the two sequences. Okay, and the same homology and as well as the similarity tools can be used to identify the degree of similarity between the novel sequences of unknown structure and as well as function. Yes, now it's time to study the role of bioinformatics in the field of plant biotechnology. Okay, so as of July 30th, 2006, scientists all around the world are pursuing 2,126 genomic projects. Already 406 genomic projects got published. There are 1,665 projects are ongoing still. Okay, so in the field of medicine, this bioinformatics means that it has wider field in which potential cure yet to be discovered for various diseases. In the field of agriculture, bioinformatics paves the way to understand the plant evolution and also to gain the knowledge and apply the same knowledge to improve the crops. Bioinformatics is a new science that combines the power of computer, mathematics, algorithms and statistics in the concepts of life science to solve all the biological problems. Okay, through bioinformatics, scientists have been able to analyze various genomes. This bioinformatics mainly deals at four different levels. One is at the biological level, secondly with the molecular structure, and third expression data, and finally this bioinformatics deals with the bibliographic data. So let's see one by one. In case of biological data, bioinformatics mainly deals with the DNA, RNA and protein sequences. Okay, it deals with the sequences of nucleotides of DNA and RNA, and it deals with the sequences of amino acids in case of proteins. So all this is possible through the laboratory sequencing methods. In the field of molecular structure, higher molecular structure can be obtained by combining thermodynamic data and computer modeling with the measurements from the laboratory methods like X-ray diffraction method and as well as magnetic nuclear resonance imaging. In the expression data, scientists use microarrays to determine when and where these genes are expressed. Okay, the same microarrays have been used to understand the complete genomic expression in certain cell types or in specific environmental conditions. If we move on to the bibliographic data, the number of scientific articles have been increased from last few decades. So this is mainly because of increase in the genomic projects and as well as sequencing programs. What can bioinformatics do with all this data? The first step making sense of all the biological sequences and structure is to formulate a method to manage the data as well as to process and to maintain it. Database management is the first and as well as the fundamental task in bioinformatics, and bioinformaticians do this by assembling the information in the databases. What is this database then? See, generally database is the collection of information and it is stored in a systematic manner. Okay, in case of bioinformatics, see, this information will be in the form of sequences of RNA, sequences of DNA and even protein sequences. And all these sequences are arranged in a systematic manner. That means according to their function, according to the species from which they have come from, or according to the journal article which have been published at the first. Hope you like this video. Please do subscribe to my channel and hit the bell icon. Also never miss an update from me. So stay tuned. See you in the next video. Until then, take care.